Hello, everyone. What we're going to talk about in this lecture is the first class of biomolecules which are referred to as carbohydrates. So what we'll go in this lecture, first we'll define what carbohydrates are and why are they called that way. Then we'll talk about their function in living organisms. What do they do there? And then we'll go through the different classes and structure of the different type of carbohydrates or saccharides. So we'll go through monosaccharides, disaccharides, and so on. And finally, we'll focus on the structure and function of all of these components. First of all, why are they called carbohydrates? And in order to understand that, I want you to briefly pause this video and go through the structure of these two examples of monosaccharides in the linear form right now, which are called glucose and fructose. Now, what I want you to do is count the number of carbons, oxygen, and hydrogen that are found in them. Now, once you unpause the video, you'll see that both of them are made by the exact same type of elements. So which are carbon, hydrogen, and oxygen. And now, if you count them in glucose, for example, you see that you have carbons, 12 hydrogens, and 6 oxygen. So hence the chemical formula is C6H12O6. Now, if you count them in fructose as well, you see that you get exactly the same chemical formula, C6H12O6. Now, you will see the same thing if you went through other monosaccharides such as some ribose, galactose, and so on. And so what the first scientists were doing exactly this. They were studying the structure or the components that were found in these saccharides. And they were finding exactly what you found now by counting the number of elements in each of these compounds here. And so what they came up with was that what they noticed is that all the formulas, all the monosaccharides that they were studying could be all written in the same way with a CnH2n, so double the number of the carbons. The hydrogen will always be the double the number of carbon. And the oxygen would have the same number of carbon in all of them. Do the same thing with ribose, galactose, you get the same thing. So they could actually simplify this formula, I should say, in a formula like this, which basically you can translate into a hydrated form of carbons. So N carbons hydrated with N number of water molecules, hence the name carbohydrate, hydrated form of carbons. That's where the names come from. In terms of function, so first of all, I used to say these are the most abundant biological molecules. You see they are used for so many things, particularly for energy storage, structure, particularly in cellulose, which is the most abundant if you want carbohydrate on Earth. And they're used also in short forms, what we call oligosaccharides attached to some proteins, which form what we call glycoproteins, are used also for signaling. So cells, they signal with each other or to the immune system using proteins modified with carbohydrates. So part of the function of carbohydrates is also in the signaling. Okay, in terms of classes, now carbohydrates are classified based on the number of the basic monomers, which are referred to as monosaccharides. So a single molecule here, a saccharide, is a saccharide, we're referred to as monosaccharides. Glucose, ribose, galactose, fructose, and so on are monosaccharides. So based on the number, if you have only one of them, we would talk about monosaccharides. If you have two of them joined together, we're talking about disaccharides. If you have a few of them, now the number changes, but from 3 to 10, I would say, you can talk about oligosaccharides. This number you can find a variable depending on the scientist you talk to. But just few of them, talking about oligosaccharides. Now, if you're talking about hundreds of these monosaccharides joined together, at this point, you're talking about polysaccharides. They can go up to thousands. Glycogen can have 15,000 glucose molecules joined together, just to give you an idea of a polysaccharide. Now, first type, monosaccharides. Now, monosaccharides are divided in subclasses based on the number of carbons that they have. This number of carbons can vary between 3 to 7. So if you have a monosaccharide, such as glyceraldehyde, which contains 3 carbon, you talk about triose. Tri means 3. If you have one with 4, you would talk about tetrose. 5, such as ribose here, has 5 carbons, you talk about a pentose. 6, such as glucose, mannose, see here in the pictures, fructose, they are hexes because they have 6 carbons. And finally, if you have 7 of them, you talk about heptose. Now, structurally speaking, you could probably already see they all contain some, I forgot to put a slide for that, but they all contain, so a carbonyl can like in this case, all these 3 cases, or if you get back to fructose, they other monosaccharides, they contain a ketone group, so a carbonyl groups of the type of ketones. Together with that, they're extremely rich in hydroxyl group together with them. Now, talking about isomers. So now, if you compare different monosaccharides, as we compare at the beginning, trying to explain why or where the name carbohydrates come from, we compare glucose to fructose, and we came out with the same chemical formula. So now, compounds that have the same chemical formula, but their elements are arranged in different ways, they're referred to as structural isomers. Again, glucose and fructose, they have the same chemical formula, but if you look at the structure here, they're different from each other. So they are defined as structural isomers when they're compared to each other. Now, even within the same type of molecule, you may actually have different versions. And in this case, what we're talking about here, we're talking about optical isomer. So now, these molecules here, taking some time here. So each of these carbon, because of the angles you have, can have actually some of the elements put in different orientation. So these are referred to as asymmetric carbons, okay? So what it means is that these groups here can actually distribute slightly different in the same molecules, forming what we call isomers or enantiomers type of thing. So in general, molecules that can basically give origins to 2 mirror image of each other, which cannot be superimposed, are referred to as optical isomers or enantiomers. So these are same type of molecules. So we're talking about, for example, glucose. Glucose can give origin to molecules that are basically mirror image of each other, but they cannot be superimposed. And so can give origin to optical isomers or enantiomers, as you can call. So, and these have to be superimposable. For example, look at these 2 examples right here. On the left side, now you have pair of glasses here, which you put in front of a mirror. This is going to be the mirror image you see. Now, if you take the 2 image, these glasses can actually be superimposed to each other. So these are not considered enantiomers. These are not optical isomers. They're identical form of the same. Now, instead, if you change one of the lenses, okay, to a color one, so one doesn't have any color, the other has it, and now you put it in front of the same mirror, at this point, the 2 mirror image that you have will not be superimposable anymore. The same thing happens if you look at your 2 hands. They're basically a mirror image of each other, but you cannot impose them to each other, superimpose. So they will consider as optical isomers or enantiomers. So to be an enantiomer, the 2 mirror image are not, superimposable. They're not superimposable, I should say. Sorry, there is a mistake there, so not. Let me just write it here so everyone is clear with it, not superimposable. Okay, so I'm writing with the mouse, not the best one, but are not superimposable. Now, there are different molecules, so applied to monosaccharides. Glyceraldehyde is another molecule. You can see how it works. So this is, you see it on the bottom here. This is glyceraldehyde. So this carbon here, it's a chiral center. That's what you would call it. And so the groups attached here can be in different orientation, creating two forms, one referred to as the D or dexter. D comes from the Latin word that means right in English. So D, dexter. L stands for levis, which means left in English. This is an older way of classifying, by the way. So these two forms now are mirror image of each other, considering this carbon here, and therefore they're not superimposable, okay? So they're considered an enantiomer. The same thing for glucose. Glucose 2 exists actually in different forms. Each carbon can be a center of chirality there, and therefore there can be different enantiomers there. But the main forms are these D-glucose here and L-glucose, with the D-glucose being the most abundant form found in nature. So it can exist in different forms, DL, but these two forms, first of all, are very different biologically speaking. So enzyme will recognize only one or the other forms. And also one of the forms is more abundant than the other. So the D-glucose form, for example, is more abundant than the L-glucose. Now, until now, I showed you monosaccharides in the linear form. In reality, saccharides, monosaccharides, did exist 99% in the cyclic form. And the cyclic form is formed through a reaction intramolecular. So within the same molecule, there is a reaction that takes place that causes the formation of a cyclic ring. Now, what I'm telling you right here applies to glucose, but the same thing applies also to other molecules such as fructose, galactose, ribose, and so on. So to any monosaccharide, ultimately that has 5 to 7 carbons, so the maximum number. So a triose and a tetrose could not form the cyclic form. So how is this cyclic form formed? Is you can see here an example for glucose. So what you have is, if you remember when we talk about the functional groups and we talked about carbonyl group, I told you the carbonyl group, whether they are aldehyde or ketones, in this case is an aldehyde, they're very reactive. And that's exactly an example of it. So this group here is very reactive, and it will react with the hydroxy group attached to the carbon 5. So what this leads to, and you don't need to remember all these details, by the way. So what it leads to is there's some electron transfer from this hydroxy group to this oxygen, with the proton as well being transferred here. Basically leads to the conversion of this oxygen here into an hydroxy group, so an oxygen and a hydrogen attached to it, OH, and the formation of an oxygen bridge ultimately between the carbon 1 and the carbon 5. So this is the carbon 5 that was here. This is the carbon 1 that was here, okay? And this oxygen here has been transformed into an hydroxy group, so OH, that you can see here on here. And then I'll tell you in a second the orientation. So this leads to the formation of a cyclic form. Basically, this looks like a plane, okay? This is a Howard projection of the same molecule here, so it gives you a little bit more three-dimensional idea. So here is thick as like if it's coming out from the screen. Now, an important thing to understand is that whenever a monosaccharide with 5 or more carbons becomes cyclic, now the hydroxy group that is formed in what used to be the, in this case, aldehyde group, so the carbon group, can end up in two possible orientation compared to the plane, this plane here. It can end up below the planes, so imagine this being coming out to you. In this case, this hydroxy group will be below, or it can end up upward on top of it. Based on how it turns out, so if it ends up going below the plane, you talked about the alpha form of that monosaccharide. So in this case, we're talking about alpha glucose. If it goes upward, it will be referred to as beta glucose. Now, is this important? Yes, it is. These, biologically speaking, are two completely different molecules. Even though they're both called glucose, they're two completely different molecules. This is the monomer that makes up, just to give you an idea, polysaccharides such as amylose, amylopectin, glycose. This makes up cellulose, all right? So with completely different function. This is used for energy production and storage. This is used for structural purposes. So very different molecules recognized also by different enzymes as well. Now, what are the main monosaccharides? And I want you to remember the structure of all of them. There are 2 pentases, so 5 carbon monosaccharides I want you to remember. And the reason why I want you to remember them is because these are extremely important in the formation of the nucleotides that give origin to DNA and RNA. So one of them is, I should start with ribose, it's the one right here. And the other one is a version of ribose that is missing an oxygen here, and therefore it's called deoxyribose. Both of them are pentose, so they have 5 carbon. 1, 2, 3, 4, and 5. Very similar. These minor differences is huge when it comes to nucleic acid. The other 3 monosaccharides I want you to remember, because these are key components of the disaccharides we'll talk about in a second, and the polysaccharides we'll talk about at the end, are galactose, glucose, and fructose. When it comes to memorize their sequence, I highly recommend that you memorize them by comparing them to each other. For example, if you compare glucose to galactose, so if you memorize one of them, glucose, I remember this pattern down, up, and down in terms of hydroxy group, and this is in the alpha form. Now, if you compare them, the only difference, forget about the alpha and beta, this is beta glucose, but it could have been in alpha if you want to, it's in the hydroxy group here. So this is up instead of down. Everything else could be the same, plus minus the alpha and beta configuration. One other thing I want to stress is that all of them, since they become cyclic, they can exist both in the alpha or beta configuration. Next level, next class of polysaccharides, or sorry, carbohydrates are disaccharides. Disaccharides are molecules that are formed by 2 monosaccharides joined together. Now, they're joined together through a dehydration reaction, if you remember. So you have, in this case, 2 hydroxy group, I'll show you in the next slide, reacting with each other, forming a water molecule. And the bond that is formed, in this specific case, is called glycosidic bond. The glycosidic bond is actually referred or specified using 2 important information. The first thing is by telling what type of form you had in the first monosaccharide. Is it in alpha or in beta? And the second thing, the second information you specify in the bond is the number of carbons that are involved in the formation of the bond. Let's go through some example here. So this is a glycosidic bond between 2 glucose molecules. This is what makes up maltose. Glucose and glucose. Now, if you imagine them separate, so imagine before the formation of this bond here, this glycosidic bond, the hydroxy group here, since the hydrogen is upward, the hydroxy group here would have been on the downward side, so this would have been alpha glucose, and so was this one. But the relevant one for the naming of the glycosidic bond is the one on the left side, because it's the one that contributes the hydroxy group for the alpha or beta. So this was in alpha. So this bond is defined as an alpha. And now it's between the carbon 1 of the first glucose and the carbon 4 of the second one. So this is referred as alpha 1, 4. Here you can put a comma if you want instead of an arrow. So alpha 1, 4 type of bond. Now, the type of bonds that you get is extremely important. It makes a huge difference. Just to give you an idea. So these are polymers. So cellulose is a polymer of beta glucose, and this is how it will look in starch and glycogen, part of the structure there. Now, cellulose is made by beta glucose with a beta 1,4 type of bond. Whereas starch and glycogen they have alpha 1,4. So alpha glucose joined together with an alpha 1,4 type of bond. Notice the difference. So in this case, because they are in beta, the planes of the different monosaccharides, which are part of these polysaccharides, they end up being in a fairly linear straight line. So they give origin to the linear structure. Whereas when they are in beta, because of the orientation of that hydroxy group, which then ends up glycosidic bond, now this gives origin to a turning type of a curve structure, which then turns around. Okay, there is a big difference between an alpha and a beta bond, ultimately in terms of structure, which correlates also to the function. This is a structural molecule. This is an energy storage, a glucose, and therefore energy storage type of molecules. So the fact that the curves allows for compacting more molecules per unit of space than if it were linear. Okay, let's go through the 3 disaccharides I want you to remember here. So the first one is sucrose. Sucrose is a disaccharide formed by alpha glucose joined to beta fructose. Now, this is formed through a hydration reaction here between the carbon 1 of the alpha glucose and the carbon 2 of the fructose. Notice that your book has the wrong structure and bond. So make sure that you use this one to identify sucrose if I ever ask you on a test. So the joining together is between the carbon 1 of this alpha glucose and the carbon 2 of this fructose molecule. Hence, this bond is referred to as alpha 1,2 glycosidic bond, which gives us sucrose, which is what is actually your table sugar, the one you use maybe on your coffee. Second important disaccharide is maltose. This comes from the degradation of starch broken down into dimers, so which are maltose. Maltose is built by 2 identical monosaccharides, which are alpha glucose. So this is an alpha glucose. It's an alpha glucose joined together with a bond, glycosidic bond between 1 and carbon 4. Hence, this is referred to as alpha 1,4 glycosidic bond. Third disaccharides to remember is lactose. That disaccharide is abundant in cow milk, which is the source of problems for people that do not have the enzyme that actually degrades it. The lactase breaks this type of bond right here. Some people do not express it, hence they are lactose intolerant. So what is it formed by? It's formed by 2 monosaccharides, which are beta galactose. If you see here, the hydroxy group would have been upward, so beta galactose, and glucose here, alpha glucose here. So the bond that you have in there is between carbon 1 and carbon 4 of galactose and glucose, respectively. It's in beta, so it's a beta 1,4 type of bonds that you have in there. Okay, the last class I'll talk about here is polysaccharides. So we skip the oligosaccharides. Oligosaccharides are short chains of monosaccharides. In my definition, between 3 and 10. Some other people use less, 3 and 6, or take it as 3 and 10. When you start talking about hundreds and thousands of monosaccharides joined together, at this point you're talking about polysaccharides. So these are polymers, all of them, you see, of glucose or modified forms of glucose, like when we talk about chitin at the end. So these are the 4 main polysaccharides we'll talk about. In reality, it's 5. We'll talk about starch, which is made by 2 polysaccharides, amylose and amylopectin, glycogen, cellulose, and chitin. Starting with starch. So actually, before we get there, so polysaccharides can be either linear, such as amylose, cellulose, chitin. They are all linear, so they don't have, or they can have branches. So meaning that they're formed by a linear chain, like this one, which at times projects or joins other linear chains, forming branches, similar to what you will find in a tree. And the amylopectin and glycogen are basically almost identical. and they are branch type of polysaccharides. So the difference between linear and branch, as mentioned before, is in the type of glucose that is found in them, and therefore the bond that is formed in there. So cellulose is formed by beta glucose. The bonds are beta 1,4, and therefore these molecules turns out to be linear. And in starch and glycogen, so chitin would be similar to cellulose. Starch and glycogen, the bonds, the main bonds, is all made by alpha glucose through alpha 1,4 bonds, and this gives origin to circular molecules. Starting with starch here. So this is found in plants where it is used to store glucose and therefore energy. Starch is formed by alpha glucose. In reality, exists in two types. There is amylose, which makes up about 20 to 30% of starch, and the second form, which is amylopectin, which makes up about 70 to 80% of starch, depending on the plants. Now, the difference between them is that amylose is linear, whereas amylopectin is branched. What do we mean by that? So amylose, it's a linear polymer or alpha glucose. It's basically maltose attached to maltose molecule. So imagine that. So this will be alpha glucose attached to alpha glucose, all attached with an alpha 1,4 glycosidic bond, like in maltose. Okay, you can get about 4,000 units attached to them. They curve, so they can compact fairly well. Now, amylopectin, if you want, is a more evolved version of an energy storage molecule because it's like amylose. Amylose has the same basic linear structure, which, though, at times joins with other linear structure through an alpha 1,6 bond. So a bond at this point between the last carbon in another linear structure, so carbon 1, which is in alpha, and the carbon 6 of a glucose molecule, which is of another linear structure. So imagine these two of them here, from time to time being joined and therefore forming branching. So this, I say, it's more evolved because through the branching, you can actually compact even more glucose and therefore more energy per unit of space. Similarly, glycogen is the polysaccharides which is instead found in plants with the same purpose, to store glucose and energy. Its formation, it's induced in skeletal muscle and liver when insulin is produced by your pancreatic cells. Like amylose and amylopectin is formed by alpha glucose. So it forms long chains with alpha 1,4 bonds that curve around, and like amylopectin is basically identical to amylopectin. It has branching with alpha 1,6 glycosidic bonds between the last glucose molecule of one chain, so this will keep going, and another glucose molecule in between chain. The difference between amylopectin and glycogen is that this branching here is more frequent in glycogen compared to amylopectin, making it a little bit more efficient to store energy. This is a picture that shows you, this is actually a protein that's the center of a glycogen molecule, to show you how glycogen is actually stored and branching out and storing, therefore, a lot of glucose molecules in your liver and skeletal muscles. Third polysaccharides we talk about here is cellulose. So this is a structural component that is found in plant cell wall. So makes the cell wall of plants extremely, extremely strong. Okay, this is the most abundant polysaccharide found on Earth. With all the plants that you have, you can easily imagine why. And now it's formed by a linear unbranched fibers of beta glucose. Instead of using alpha glucose, it's made of beta glucose up to 15,000 units you can find altogether, which do form a linear structure through beta 1,4 glycosidic bond. Being beta glucose, they form beta 1,4. So this is projected up, is in beta, and this is carbon 1, carbon 4. So all of these are beta 1,4 bonds forming this polymer of beta glucose, linear. Now, this is normally digested by an enzyme called cellulase, which unfortunately we cannot produce, and that's the reason why you cannot really digest easily grass and stuff like that. I don't know about you, but I can't. Now, how do these cellulose molecules give strength? Is by working in large numbers. So in reality, if you look at the cell wall of a plant, you don't have singular fibers, but you have thousands of fibers. So each one of these, horizontally speaking, is a fiber of cellulose. This is cellulose, 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 and cellulose. And they get strong together by forming hydrogen bonds here in between the fibers. So if you remember, this was an example that I used when we're talking about hydrogen bonds, saying that they were very, or 1/10 to 1/20th in strength compared to a covalent bond. So very weak, but they work with large numbers. And this is an example. Here you end up having millions of bonds here that make up a super, super, super strong structure of cellulose fibers combined together. Last polysaccharides we'll talk about here is extremely similar to cellulose. It's chitin. This is the polysaccharides that makes up the exoskeleton in insects and crustaceans, like this crab right here. As you know, try to bite at least a crab, you know how hard this is. And this is because of the presence of these polysaccharides, chitin. So it's extremely similar to cellulose. Imagine just cellulose, but instead of being made by beta glucose, it's made by a modified version of glucose called acetylamine, amine glucose, N-acetylglucosamine. I'm sorry. Is that the end of the lecture? I'm almost done. N-acetylglucosamine here. So that's the difference between glucose right here and N-acetylglucosamine, which is found in chitin. The type of bond is exactly the same. Beta 1,4 bond. Again, the monomer, the difference is that the monomer is different. There is formation of more hydrogen bonds in between, hence even a stronger structure compared to cellulose. Okay. So in the end, summarizing every talk about here. First of all, we define why carbohydrates are called carbohydrates and what their function is in living organisms. Then we talk also about the bonds that joins them together, the glycosidic bond. We define how they are named, what their type they are, and so on. Then we talk about the different classes of carbohydrates, and we talk about their function and structure as well. So we talked about monosaccharides, focusing on ribose, deoxyribose, glucose, fructose, and galactose. We talked about also defined isomer, optical isomers, and so on, structural isomers. And then we talked about how the cyclic form is produced, starting from linear form, giving origin to an alpha or beta forms of different monosaccharides in the cyclic form. Then we move to disaccharides, focusing on the structure of sucrose, maltose, and lactose. And then we finish by talking about polysaccharides. So we looked at the difference between them, between the linear ones and the branch one, their function of energy storage and structural function. And then we focus on these polysaccharides. Starch, which is made by amylose and amylopectin, then glycogen, and finally cellulose and chitin.